Nafish Wadhwa. Nafish and his family joined our department this year, and also Mike Lynch Center for Mechanism of Evolution. Uh, Nafish did his undergraduate at IIT Delhi, and then he earned his master degree in fluid mechanics from Virginia Tech. And then he switched to biophysics. He did his, I'm sorry, I forgot. He did his PhD in Denmark. And then he followed that with a postdoc in the labs of Howard Burke, which is the giant in bacterial motility, and co-supervised by Ethan Garner. So this is fantastic. It's a combination of a young, hungry faculty and basically the father of the field. Like many of us, Nafish received many awards, and I invite you to read his CV. One of them is K99 from NIH. For non-biologists in the room, this is very prestigious, like elite cohort. The name is K99 Pathway to Independence. One thing that caught my attention in his CV, sorry that I'm giving a long introduction here, is that Nafish also participated in 5-week workshop on inclusion in STEM education. So ASU is the right home for people like Nafish. And that includes training on managing labs and lab dynamics. So we expect a lot from your lab, Nafish. And this afternoon, he will talk about bacterial motility. Nafish, take it away.
Okay, thank you, Rizal, for that very kind introduction. I'm really glad to be here and be giving this talk on some of the ideas and questions that I'm really passionate about. And I want to say up front that if you have any questions along the way, you can just raise your hand and ask me those questions. I would much rather have a conversation than be giving a lecture. All right, so let's start. So the question for today's talk is, do bacterial engines have automatic gear shifts? And this question itself gives rise to several sub-questions. First of all, do bacteria have engines? Why do they have engines? And okay, they have engines, and what are these automatic gear shifts that we're talking about? So I'm going to try and touch upon all of these questions in today's talk. And my goal really is nothing more than that you leave from this room with a little bit increased appreciation towards little organisms like bacteria. That is all. Okay, and the engine that we're going to talk about is very beautifully represented by an artist, Dan Nowakowski, based on very recent data that have been collected in the field on the structure of these molecules, structure and dynamics of these molecules. So we're going to talk about these things. Now, to start off this conversation, I want to actually think about machines. So one of the biggest achievements of human civilization is the invention of machines that do our work for us. And one example of a machine is this car. If we want to move from point A to point B, we can just sit in the car and we do not have to apply much muscular power to do this work. The car will do the work for us. Now, just like humans use machines to do their work, cells also use molecular machines to do their work for them. And I have 3 examples here highlighting the kind of work that is going on inside cells all the time. So we have this a machine on the left. It's called an ATP synthase, and it generates ATP by making these rotations. And ATP is essentially the energy currency inside all cells. So the rotation of this machine is providing ultimately the energy required to do any kind of biological function. We have ribosomes. This is another machine that generates proteins inside cells. You might know that most of our bodies, most of the structural elements, many of the functions are carried out by proteins. So this machine is also very important. And finally, we have the replisome. This is a machine that replicates DNA, which is really the basis of all information carrying in biology. So cells use these machines to carry out their tasks. Now, if you think about, again, human machines, you, for better or for worse, many of the man-made machines are these days very so-called smart machines in the sense that they can sense their environment and adapt to these changes in the environment on the go. So cars have all sorts of sensors. They can sense, for example, something coming from the front, and they can apply brakes for you. The question is, do cells have smart machines too? And I will try to convince you today, especially in the context of this one machine that we're going to talk about, that it is indeed a smart machine, that it actually adapts to changing environments. Okay, so with that in mind, let's talk about the organism of choice that I have been studying for the past 6+ years. And this organism is the humble bacterium E. coli. Now, you may have heard of E. coli because it causes horrible infections, but luckily, most strains of E. coli are completely harmless. We use them in the labs, in biology labs, on a regular basis. E. coli is about 2 micron long and a little over half a micron wide oblong-shaped cell that inhabits the guts of many mammals, including us. Now, one of the things that you notice with E. coli is these wavy filaments that are sticking out of the cell in different directions. And these filaments are really one of the focus of our talk. These are the flagella that E. coli uses all the time. And you can see that it has multiple flagella per cell. Okay. As I mentioned, E. coli has been used in biology labs for decades. In fact, if you count the number of Nobel Prizes given for biological research over time, studies in E. coli have probably won the most number of Nobel Prizes in history in biology. So E. coli is really interesting that way. But when I started in this field, I didn't know anything about all of that history. I only cared about E. coli because it swims. So if you look at E. coli under the microscope, you will see this vigorous swimming activities. All of these cells are out and about. They're going somewhere. And these bacteria, like E. coli, are in fact the smallest self-propelled swimmers found in nature. Each one of these cells is moving at speeds of roughly 10 to 20 times its body length per second. And if you try to translate that to a human scale, that would be equivalent of running or swimming at 60 miles per hour. Incredible speeds at which these cells are moving. So how are they doing it? And that brings us to these flagella that we just looked at. So what E. coli does, that it brings all of its flagella together in this bundle. So it brings all of them behind itself in a bundle, and it rotates these flagella, rotates the bundle to create a thrust, hydrodynamic thrust, that balances the drag on the cell body to allowing the cell to move forward. And so this video, the flagella of the cell have been labeled with a fluorescent dye, so we can see them. Normally, they are too small to see by a fluorescent microscope, by a regular microscope. So we can see that these flagella are basically moving like a propeller. And it's this rotation of this propeller that is moving these cells forward. Okay, that explains how the cells can move forward in a straight line. They run a propeller and they move. But you might imagine that the cell cannot just keep going in a straight line. It also must change direction sometimes. Otherwise, it'll run into a wall and never be able to leave, right? So how does the cell change its direction? It changes its direction by changing the direction of rotation of one or more of its flagella. So if you look at a series of steps here that I've highlighted, what the cell is doing here is it's doing a process called tumbling. And tumbling is the following. In the first frame, It's just moving in a straight line, rotating all the flagella counterclockwise when looked from the outside. And then in the second frame, one of these flagella changes its direction from counterclockwise to clockwise. And the result of this change in direction is that instead of rotating in a very coherent bundle, this flagella now so moves out of the bundle. And in fact, the bundle pretty much falls apart due to this kind of a change in direction. And because now the bundle has fallen apart, the cell does not have a coherent bundle to push it in one coherent direction. As a result, the cell stays in one place and just changes it moves in sort of a random motion called tumbling. And that's the motion that allows it to change direction. So after some time, the cell then reverses the direction of rotation of this rogue flagellum. Everything goes back to counterclockwise. The bundle reforms and the cell moves forward in a new direction. So by doing this bidirectional rotation of its flagella, the cell is able to move in different directions. Any questions so far? Okay, so we have seen that this rotation of its filaments drives the cell forward. Now, I'm going to try and address 3 different questions in the remainder of my talk for the next half hour or so. And those 3 questions are the following. First question, why do bacteria swim the way they do? I mean, it's a pretty peculiar way of swimming by rotating these propellers. So why do they swim the way they do? Second question, how are bacteria able to swim the way they do? What is actually energizing this motion? What is driving this propeller? And the third question, how do these bacterial engines cope with changing conditions? So I'm going to try and answer these 3 questions. Okay, let's take the first question. Why do bacteria swim the way they do? And to answer that question, we have to start pretty basic. So let's go at physics of swimming 101. If an object, any object is moving in fluid, there are essentially 4 parameters that describe its dynamics. Those 4 parameters are the size of the object, A, the speed of swimming, V, the density of the fluid, rho, and the viscosity of the fluid, eta. I should mention that this kind of formalism was not really formalism. This kind of description was really pioneered by Ed Purcell in a classic paper in 1977. So Ed Purcell did it so well that I am using exact same description. And Ed Purcell, in fact, is the same guy who won the Nobel Prize for discovering NMR. So he started his career thinking about problems like NMR, but later on, he got more and more interested in microbial swimming. So when we have this situation, we can write down a very simple scaling relationship, which is that the inertial forces that want to keep this object moving, so mass times acceleration, those kind of forces, divided by the viscous forces which want to stop this object, the ratio between those forces is given by this factor here. So it's the size multiplied by the velocity multiplied by the density divided by the viscosity of the fluid. And it's a non-dimensional parameter that is known as Reynolds number. So it's been defined as Reynolds number is defined as this non-dimensional parameter. So we have talked about how Reynolds number controls the relative importance of these forces. And this Reynolds number really impacts the lives of organisms, any kind of organisms swimming in fluid, because its value varies wildly between swimmers of different sizes. So let's think about what the Reynolds number is for a human. So for a human, the size is about 1 meter. Let's try to guesstimate it. Size is about 1 meter. Its swimming velocity is also, let's say, approximately 1 meter per second. That's the general scale we're talking about. And the density of water that it's swimming in is 10 to the 3. And viscosity of water is 10 to the minus 3. So if you plug those numbers in, we are making a very rough estimate. You would get a number that's actually closer to 10 to the 6. Ed Purcell, in his calculations, made a much more conservative estimate, and he came up with a number of 10 to the 4. So Reynolds number for a human swimmer, Ed Purcell said, is about 10 to the 4. The point is that this number is much larger than 1, which means that when humans are swimming, viscosity does not play a big role. It's the inertial forces that are important. And then if you do the same calculation for the humble bacterium, its size is about a micron. It swims at about 30 microns per second. Or size scale is about a micron. And if you plug those numbers in, you would get a Reynolds number, which is 10 to the minus 5. So much, much smaller than 1. Because Reynolds number is much smaller than 1, you can safely say that for the life of a bacterium, inertia is irrelevant. It's the viscous forces that dominate its motion. So let's take this a step further and see how Reynolds number affects the fluid motion. And to do this a little bit more precisely, let's start by looking at the equations of motion of fluid. These are the famous Navier-Stokes equations, which still remain unsolved. There is no analytical solution to these equations. And there is, in fact, I believe, $1 million prize for people who can come up with the solution. But nevertheless, we're gonna actually make some approximations that make them much more tractable. So what are these equations? This is essentially Newton's second law for fluid motion. On the left side, we have the mass times acceleration like term, and on the right side, we have all the forces that are acting on it. The first term here is the local acceleration. If the velocity is changing with time, you will get a non-zero term here. The second term is the so-called advective acceleration. As you're moving through space, if a fluid packet is moving through space and its velocity changes, that is an acceleration that we also have to account for, known as the advective acceleration. The third term here is the pressure gradient, basically the forces that are driving this motion. If you have a fluid packet, if more force is acting on one side than the other side, then it's going to try and move that way. So this is the pressure gradient. And the last force is the viscous drag, which is trying to resist the motion, the viscosity. So we have these forces. We have this equation. What we can do next is actually non-dimensionalize this equation by scaling all of our relevant terms by the typical scale that they have. So when we do that exercise, we can replace this velocity by a scaled velocity, which is scale it by v. We can scale the pressure by this term. And what we will find is that we end up with this equation where the left two terms are both multiplied by the Reynolds number. This is the same Reynolds number that we just talked about. And the effect of Reynolds number being so small, much smaller than 1, is that you can simply ignore the left side of the equation, and you will be left only with the right side of the equation. So when you do that and come back to sort of dimensional equations, you're left with this very simple equation where we have basically removed all of the more complicated terms. And this equation is called the Stokes equation. Stokes equation governs fluid motion at very small length scales. So you can notice that it's a linear equation, and it has some properties, 5 properties that are significantly that are discussed in the field. And for our purposes, I'm just going to talk about the first property, which is reversibility. Now, if this equation has a solution P and V, then you can very quickly see that minus P and minus V is also a solution. So if you reverse the motion, if you reverse the motion of an object moving in this field, you will reverse the velocity field, you will reverse the pressure field. So that's the reversibility condition, which prescribes that any kind of reversible motion of a swimmer will lead to no net motion, net displacement. So essentially, if I'm a swimmer and I'm trying to swim at low Reynolds numbers, if I do this, I will move a step forward, maybe. But because the equations are reversible, as soon as I do this, I move a step back. So if I'm doing reversible motion, I'm just going to go back and forth in the same place. So that's a property of Stokes flows, that reversible motion leads to no net displacement. And this was actually very memorably called the scallop theorem by Ed Purcell. It's called scallop theorem because a scallop, when you think about its motion, it has only 1 degree of freedom. It can either open or close its shell. And when it opens, it might move forward. When it closes the shell, it moves backwards. So this is the scallop theorem at low Reynolds number. An organism like a scallop, which can only open and close its shell, will not be able to swim. So how do actual real organisms sort of fix this constraint? How do they get around it? They get around it by using irreversible motion. And 2 of the very common strategies of swimming at small scales are rotating propellers and flexible oars. So on the left, we have our friend, the bacterium, and it is able to move because it's swimming by rotating these helices, and there is no reversibility in this. These helices are basically propagating waves, and essentially it does not reverse its motion, so it's able to move forward. And on the right side, we have this many flagellates swim by so-called flexible oar strategy. It's essentially doing the same motion that I mentioned, but its oars are very flexible. So on the backstroke, they actually kind of fold. And that's how they break the reversibility and they're able to move forward. Okay, so these are the 2 strategies. And this actually tells us why bacteria swim the way they do. Okay, so having talked about the first question, why bacteria swim the way they do, I'm going to move on to asking the next logical question: how do bacteria swim this way then? So how is this motion, this rotation, being generated in bacteria in the first place? So to answer that, you have to take these filaments and you have to zoom in at the base of the filament. So if you zoom in right over here, what you will see is that there is this beautiful macromolecular machine that is driving the rotation of these filaments from the base. So the filament itself is completely passive. There is no activity in the filament. It's just a rigid helix. But at the base of this filament is this motor that is constantly driving the rotation. This is the engine that we are talking about. So what does this engine look like? Just like any electric motor, this bacterial motor has all the same parts that an electric motor would have. So it has a rotor labeled here in yellow that rotates with respect to the cell, and it has a stator labeled here in orange that remains stationary with respect to the cell and applies forces on the rotor to drive this rotation. And it has a drive shaft. So this purple part here is the drive shaft that transmits the torque from the rotor to outside of the cell. And it has this flexible hook, which acts like a universal coupling that transmits, that allows the torque to be transmitted from this direction to this direction, wherever the filament happens to be. So you need some kind of flexible coupling to transmit this torque. And the force is being generated by the stator, which itself is actually made up of smaller protein complexes known as stator units. So the stator is a complex of these units, each of which is supplying part of the force. And before I show you that, I just want to show you this atomic resolution structure that has been found of these motors because of the advances in cryo-EM in recent years. So by now we know pretty much all of the components of these motors down to the atomic resolution. And what I brought with me today is actually a much older 3D printed structure, and I will be happy to pass it around, starting with Rizal, if people want to take a look at it. Okay, so we have this motor, and the stator is generating the force. What does the stator look like? If you take a cross section right about here, you will see that the stator is made up of multiple of these stator units, each one of it which is supplying part of the force that is required to drive the rotation. So essentially, all of these add up to resulting in the net force or the net torque. And the most conditions, you will have the total rotation speed of the flagellum will be directly proportional to the number of these units that are driving it. And this is an important concept that we will come back to. This structure, the stator, is also a dynamic structure in the sense that the bound units that are working at any time, they can actually leave and they go into a pool of stator units in the cell, and free units from the surroundings can take their place. So there is this constant turnover going on. So with this system at hand, and actually very recently, we found out how these individual stator units are functioning, which is that they're smaller rotary motors themselves. They use proton motive force as a energy source. So the passage of protons from outside to the inside of these stator units, they drive rotation. And this rotation is coupled with the rotation of the rotor. So it's essentially a gear train. There are these smaller gears, the stator units that are rotating, and they're coupled with this larger gear that is rotating the filament. And if you put everything together, it actually also really nicely explains how this motor is able to change direction. Yeah. Yeah, so there is a high concentration of protons outside the cell, and inside the cell there is a lower concentration of protons, leading to this chemical potential that is part of the proton motive force. Yeah. So this mechanism, rotary mechanism, also really nicely explains how the motor is able to change direction. So what is happening is that in the clockwise situation, this part of the rotor that engages with the stator units is in sort of a narrower position. So the rotation will then drive the motor in a counterclockwise manner. But when the motor switches, this part opens, and the same gears now engage, instead of the outside of the ring, they engage on the inside of the ring. And it's a little bit hard to see. So I have an animation here. So what you see is that initially these units, I'm going to turn off the lights for a moment. Yeah. So when the ring here is in its wider position, then they engage from the inside. So they drive the rotation in the same direction. But when the ring changes its structure, now it becomes narrower. So the same units are now engaging on the outside of the ring, and they drive the rotation in the opposite direction. So just by changing the structure of the ring that is inbuilt into the motor, it's able to change its direction. Okay, so with that, we have covered a lot of ground on how bacteria swim the way they do. They use this motor, this bidirectional motor, which has these mechanisms built in to change direction and to generate rotation. Any questions about that? Yeah. Yeah. They stay in a pool in the inner membrane. Yeah, but we don't know whether that they stay nearby or if they float away. Yeah. It's 50 nanometers. Yeah, thanks for that question. I should have put a size bar, scale bar. Yeah, it's about 50 nanometers in size, the diameter of the motor. Yeah. Yeah. So
it seems like they don't have much control over counter direction switching when they turn one of the
yeah.
I assume they have control over how long they run it that way.
Yes, that's a great point. So what they're doing is that I didn't talk at all about chemotaxis. So they're using this changing directions to actually move towards favorable or move away from unfavorable locations. So they have this whole signaling pathway known as the chemotaxis pathway. So they're controlling the concentration of this, this molecule that was shown here that binds or unbinds. So this pink molecule. So they're controlling the cellular concentration of this molecule. And by controlling the concentration of that molecule, they're able to control the switching rate. Yeah. Result.
You just answered most of my questions. So I didn't see the pink molecule.
Yeah. So that's this molecule here. So this is without the molecule, and this is with the molecule. When the molecule attaches, it forces the conformation to change from counterclockwise to clockwise. So to control the rotation, the cell just controls the concentration of this molecule. Yeah. Okay. So the next question, the last question that I want to address in the talk today is how does this engine that drives bacterial motility, how does it cope with changing conditions? So what kind of conditions might it encounter? And what I'm really interested in is mechanical conditions of its environment. I'm really interested in mechanics. So the question that I'm going to ask more specifically is how does the flagellar motor cope with changes in mechanical load? So you have this engine that driving the rotation of these flagella against the external world, and the external world might pose many different conditions for the rotation. So it might be swimming in low viscosity versus high viscosity. It might be swimming freely, or it might certainly find itself in a crowded environment like our gut, right? So these are very different mechanical conditions for the cell. How does this tiny motor, 15 nanometer motor, cope with these changes? So we're going to go back to our car and think about this problem. How does a car cope with changing conditions? So let's imagine you're driving this car and you're just going down a flat road. And you know that in this condition, the engine has to supply a relatively small amount of power or torque to keep the car going. But then if you suddenly have to go uphill, there is an automatic gear shift inside most of our cars that senses this change and responds by changing gears, so which increases the torque output to the wheels, allowing the car to go uphill. Can we do the same kind of experiment with bacteria? And the answer is yes. This experiment was done about 9 years ago now by a colleague of mine, Pushkar Lele. So what he did was the following. He took a bacterial cell, he removed all of its flagella, leaving behind this very short stub of a flagellum. And then he stuck the cell down on a surface. Now imagine you're the motor, or imagine the motor that is at the base of this very short stub. This very short piece of filament doesn't really apply any load on this motor. It's just insignificant. So we are starting with a very low load on the motor. And what he did then was to suddenly attach a 1 micron size plastic bead on this stub. So what you have done from between the situation on the left to the situation on the right is increase the load quite dramatically. And in fact, you can really, this is a very dramatic change because the load actually depends on the cube of the size of the bead. So it scales quite rapidly. So it's like you were going on a straight road and suddenly you have to go uphill. How does the motor respond? The motor responds in the following way. So here we are looking at the output of the motor. So here's speed of the motor on the y-axis as a function of time. And this arrow represents the moment this bead was attached. So if you look at the speed, initially the motor rotated at just about 5 or 6 hertz. So you attach the bead, the motor rotates at 5 hertz, but then over time, the speed of the motor increases quite rapidly, and it goes all the way to 50 hertz. And this is like your car, you suddenly start going uphill, the car is really struggling, but then it's changing gears, and over time, you're able to go at a faster speed. So there is some automatic gear shift inside this motor that is allowing it to cope with this changing conditions. And what I'm really interested in is what is the physical and molecular mechanism underlying this automatic gear shift. So that's what we are interested in. And to answer this question, we wanted to do this experimentally in a way that we could really control the load on the motor. So how can we change motor load? So we asked ourselves, what would be the ideal experiment? The ideal experiment would be, let's say there's a load versus time plot. You're starting with some load, and then you would want to change the load instantaneously. So in physics, there's a special place for delta functions and step function. So we wanted a step function. We would also like to make it reversible so we can decrease or increase the load. But finally, we would also like to make it controllable so that you can study the response of the motor to different amounts of stimuli. So you can imagine that this is not an easy ask, but we were able to come up with a strategy that was able to do all of these things. And what is that strategy? That strategy is a technique called electrorotation. This is not something that we developed, we adopted it for this problem. And here's how it goes. So we have flipped the previous situation upside down. Instead of sticking the cell to the surface, we are sticking its filament to the surface. And this is, I teach physics 121, and we just talked about relative motion a few days ago. So this is an example of relative motion. If you are in the frame of reference of the cell, the filament is rotating. If the cell is stationary, the filament rotates. If the filament is stationary, the cell rotates in the opposite direction. So that's exactly what happens. You have the cell body rotating because of the torque generated by this motor. And you can imagine that this is, a 2-micron large cell being driven by a 15 nanometer size motor. This is a huge load for this tiny motor. So the motor initially has to supply a very large amount of torque, and the cell spins at a low speed. And what we do then is apply a very high frequency rotating electric field on the cell. And what this field will do is apply an external torque on this cell in the same direction as the motor. So essentially, the motor is trying to rotate the cell. You have given it assistance. So by giving this push, you take load off of the motor. So simply by turning this electric field off or on, we can increase or decrease the load on the cell, the load on the motor. Yeah. Yeah, so what this electric field does is that it very transiently polarizes, causes small polarization inside the cell, which also rotates with the electric field. But this induced dipole is not able to keep up with the external dipole because the external field is rotating at extremely high speeds. It's 2.5 megahertz. So because of this difference, there is a phase lag between the external and the induced dipole, and that leads to this torque on the cell. Yeah. 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 That's exactly right. So occasionally when you're just looking at cells under the microscope, some of them will just get attached to the surface through the filament. And this is exactly what happens in that situation. The cell will start rotating because of the motor. Yeah, great question. Okay. So importantly, we can make this load change instantaneous. We just turn the field off and on. We make it reversible, goes either way, and it's controllable because we can really control the strength of this external torque by changing the strength of the electric field. So what does this look like? It looks like the following. So every time the field is on, the cell spins at a very high speed, load goes down, and every time the field goes off, the cell spins at a low speed and load goes up. And importantly, it's only during the off periods that we can measure the state of the motor. We cannot tell anything about the motor's state while the field is on because it's being driven by this external field. So we keep that in mind when we design our experiments. So what we do in a single experiment, so I'm walking you through one of my experiments. What's happening here is that we are measuring rotation speed on the y-axis as a function of time. And every experiment has 3 phases. It has this initial phase. We're observing the initial state of the motor. And then we turn electrorotation on, decreasing the load. We keep it on for about 6 minutes. And then we turn electrorotation off, which increases the load back again. So let's see what happens to this cell. It starts with about 10 hertz or 11 hertz. So that's the speed at which the motor is rotating initially. We turn electrorotation on, but remember that we cannot observe the state of the cell while electrorotation is on. So we turn it off every 8 seconds for 1 second. And that's why you see these sparse data. And what you see is that the rotation speed of the motor actually goes down. And it goes down in what looks like steps. And the interpretation of these steps is that the motor is actually releasing these individual stator units that were driving the rotation. Because the rotation speed is proportional to the number of pound units, you see this decrease in rotation speed over time. And at the end of this phase, we turn the electrorotation field off. Now this increases the load, and the motor responds now by increasing, incorporating these stator units again. It recruits those stator units again. And you can really clearly see the stepwise increase in the rotation speed due to sequential addition of these individual units. So essentially, this is a macro scale experiment, but we are getting single molecule resolution. We can actually monitor the binding and unbinding of these single protein complexes. And from these data, we can fit steps on these data. And because we know that the rotation speed is proportional to the number of units, we can extract the number of units as a function of time from these data. And that looks like this. So in this experiment, we started with around 10 units that were driving the motor initially. And when we decrease the load, it actually goes all the way down to 0. The motor completely stopped functioning at the end of this low load phase. And when we increase the load again, it goes back to around 9 units. So what this experiment is telling us is that the stator complex of the motor itself acts as an automatic gear shift that allows the motor to adapt to changes in load. So this stator remodels at high load. The motor is driven by a full set of stator units, so which is 10 or 11. You decrease the load, the motor releases stator units, and this process is completely reversible. You increase the load again, it regains those stator units. So just by doing this binding and unbinding, the motor can adapt, be continuously adapting to its external environment. Yes?
Keep the cell on high load for a long time. How stressful is that on the cell?
Yeah, that's a great question. So if we say keep it on high load for a long time, it is not exactly stressful. The cell, if we don't do anything, the cell will keep spinning at 10 hertz for days. No problem whatsoever. Yeah. Yes?
Correlates like exactly what data would be that size and power, like
So can you repeat the question?
Oh, is there something that correlates like one data would be that much?
Yeah. So what we do know is that the maximum number of units that can be bound in the motor is 11, and that would be sort of that would correlate with the maximum speed at which it rotates. Yeah, yeah, and we can count the number of steps, and we at most see 11 steps. Yeah, yeah. Yes, and I didn't talk anything about that, so you can drive electrorotation backwards and measure sort of the torque output of the motor. And these experiments actually were done in the 90s already. Yeah. Any other questions? Rizal.
When you introduce electric field, you get dual heating.
Mhm.
So one can argue that the reason why the number is less is elevated temperature.
That's a great question. Yeah, that's a fantastic question. So temperature was our enemy in this experiment, because if temperature changes, everything changes in biological systems. So we had to build a whole sort of thing to keep the temperature stable at 20 degrees. So we are keeping the temperature very much constant at 20 degrees. Yeah. Okay, so what does this mean for the molecular mechanism? What do we currently believe about the molecular mechanism that is allowing this process to happen? We believe that the unbinding rate, the rate at which these units leave the motor, depends on the torque that they are producing themselves. So there is this feedback mechanism built into these proteins that keeps them bound if they're producing high torque and lets them go if they're producing low torque. And at high load, they have to produce high torque, so they would remain bound, and therefore the number increases. So how would that even work? So this is sort of a rough idea of how that might work. At low torque, these units we believe are in a relaxed sort of a conformation, and they can easily unbind. But when you increase the load, they have to produce a high torque, and because of this torque that they're producing, they will be physically stretched away from, they will be mechanically stretched away from the position at which they can unbind. And that leads to a reduction in their unbinding rate that would ultimately lead to increased stoichiometry. And this is actually very counterintuitive concept, because normally if you have a bond and if you pull on it, you expect that this bond will break. But here something opposite is happening. You have a bond, and you're pulling on it, and this bond is becoming stronger. And these are a class of bonds in many biological systems known as catch bonds, which become stronger as you apply force on them. And sort of a macroscopic example of such a catch bond would be if you have two hooks connected with each other. And if you're trying to release this bond, if you just pull on it, this is not going to work. You actually have to let go, and then you will be able to release those two. So this is something like that going on at the molecular scale. There are a few other mechanisms in which this could work. This is just one example. Okay, so this is really the take-home message of my talk, which is that the stator complex of the flagellar motor itself acts as an automatic gear shift that allows this engine to respond to changes in its mechanical environment. And I just want to end my talk by thanking the people who have really helped me through this journey of bacterial flagella. And most of them is Howard Berg, who Rizal mentioned was really the founding father of this field. Howard started working on bacteria when it was not popular yet. And he was the first person to show that bacteria actually rotate their flagella. And he was a leader in the field for a very, very long time. And sadly, Howard passed away last year. But I'm really grateful to Howard for taking me under his wing and teaching me everything about bacterial motility. And I also want to thank my theory collaborators, Rob Phillips and Yuhai Tu, who have been really influential in moving this work forward, and Ethan Garner for co-mentoring me during my postdoc, and my collaborator, Alberto Sassi. And finally, I also had, during my time in my postdoc, I had several really brilliant undergraduate students who did some really nice work, and they then moved on to other places. So I will stop here, and I will just go back to this slide, and I'm happy to answer more questions as the time goes. Thank you.